This is my presentation on the TARDBP gene mRNA alternative splicing protein, which is TDP-43. Here's the abstract. It's found in the cell's nucleus in most tissues. That's extremely important in its function. SMPs can cause misfolding, which is involved in amyotrophic lateral sclerosis, better known as ALS. That was what the whole ice bucket challenge was for. And general neurodegeneration. The string database shows that this protein has a strong relationship with genes such as the HNRNPH1 and HNRNPR, which both create mRNA processing proteins. And as this is an alternative splicing protein, that makes sense. And an NCBI article states that in a mouse study, embryos died after 7.5 days with a mutation in the TDP-43 gene, indicating that it has a very important function in prenatal development. The function of TDP-43. It's involved in processing mRNA. TDP-43 alternatively splices different mRNA, meaning that TDP-43 is in charge of making many different types of proteins, which explains why a mutation in it would cause such great damage. You can see in this little figure here, it shows what happens in the neurodegeneration when TDP-43 is incorrectly folded or there's a mutation in it. A PubMed search yields a mass of articles, the second most popular being an article about the possible misfolding of TDP-43. It goes into ALS, and a couple other articles have general neurodegeneration and a role in Alzheimer's. More of the function, here you can see the string database. It has a lot of intense ties to all these genes, and a lot of these genes are involved in transcription of DNA, creation of protein, and cell cycle control, which once again explains why it is a huge part in prenatal development and why it's so important to the human body. It's pretty much, present in all of our cells, and it's what helps our cells read proteins. Expression in the brain and body. TDP-43 is present in almost all tissues, as I just said. It is in the nucleus of the cells. You can see here from this diagram from the Human Protein Atlas. It has very low tissue specificity because it's pretty much all the way in your body, all around in your body. More of the expression, there's a chart. You can see on the right that these are just all the different cells in the human body, and all of them have TDP-43 in them. This is another diagram from the Human Protein Atlas. It shows that TDP-43 is present in the nucleus of cells, in the nucleoplasm. Mutations in TDP-43. I searched on GWAS and it did not yield any SMPs or risk alleles. A couple Google searches displayed one risk allele, which was the RS367543041 allele. And they say that TARDBP mutations in individuals with sporadic and familial amyotrophic lateral sclerosis, or ALS, they found that this mutation was in that. A high frequency of TARDBP genes mutations in Italian patients with amyotrophic lateral sclerosis. So a lot of the mutation or this risk allele was found in people with ALS. According to a PubMed article, the misfolding of TDP-43 due to mutation causes mass deposits of TDP-43 in tissue, and in the case of ALS, a lot of it is in muscle tissue, which is why people have trouble with motor actions in ALS. They act as inclusion bodies in the brain and spinal cords. You can see here in this diagram, it's just a diagram of what happens with this excess deposit of TDP-43 proteins in muscles. More about mutations. Mutations in TDP-43 are involved with ALS, or amyotrophic lateral sclerosis, as I've been saying. Around 97% of sporadic ALS patients, these are ALS patients that have developed ALS and it wasn't passed down genetically from their parents, or the allele wasn't passed down from their parents. Around 97% of sporadic ALS patients contain mass deposits of TDP-43 in their neuronal inclusions, suggesting that TDP-43 mutations play a large role in the development of ALS. Quick experiment. An experiment that has already been performed involved the loss of TDP-43 at the embryonic stages. This was conducted on mice. Their experiment proved that TDP-43 was essential at the early stages of life, dying 7.5 days after the embryo was formed. This is more about TDP-43's function in just creating the body and everyday life, but I wanted to do something more about ALS, especially because ALS is such a hard disease in the industry to cure and figure out why it's caused and all that stuff. So my experiment is to conduct something more related to ALS. My hypothesis, well, I mean, we'll go over the experiment first. I think the best idea is to find mice with ALS and use CRISPR-Cas9, which is probably the leading, protein or way to fix or correct mutations in proteins. So I would use CRISPR-Cas9 to correct the TDP-43 protein in the experimental group. Like, while I don't know if this is true right now, it's CRISPR-Cas9 is something that's constantly being bettered and getting a better understanding of. So I'm certain it'll be possible in the future. My hypothesis is that mice with the corrected TDP-43 gene will experience less side effects of ALS due to reduced excess deposits of TDP-43 in tissue. I don't think it'll completely cure ALS, as there's a couple other genes that can also cause ALS. TDP-43 is not the only one, but I think that it's a huge possibility to experience less side effects, or at least cure them somewhat, because ALS is a pretty terrible thing. No one can move, your muscles don't work, you have the reduced activity, all this stuff, and that's pretty much it.